right, so let's move on to our third speaker for today, which I have the honor to introduce. Dr. Augustine Doronilla is a senior analytical chemist at the School of Chemistry of the University of Melbourne. He migrated with his family to Melbourne, Australia 45 years ago, where he completed his tertiary education. He has been involved in research and implementation of ecological restoration projects in mining areas since 1988 in Western Australia, Victoria, New South Wales, and Northern Territory. In 2001, he undertook his doctoral studies at the School of Botany of the University of Melbourne on phytoremediation of arsenic-rich gold mine tailings. As a research fellow with the Environmental and Analytical Chemistry Research Group at the School of Chemistry, he has published papers on heavy metal bioavailability and transfer, in particular of arsenic, mercury, cadmium, and zinc, metal hyperaccumulation in plants, food chain transfer of toxic elements, soil chemistry, and plant nutrition. He has also done work on restoration ecology and post-mining reclamation. From 2009 through the Balik Scientist program, he has been involved in collaborations with Filipino researchers and academics, which he will talk about today. So ladies and gentlemen, without further ado, here is Dr. Doronilla.
Thank you very much, Jem. Can you hear me all right?
Yes.
Okay, so this is an opportunity for me to share of the work that I have done in my experience as a Balik scientist over the last 10 years. It's good to look back and to really appreciate these collaborations between us in different parts of the world as Filipinos to really look at the betterment and care of our local environments. So my interests are plants that are quite unique. These are plants that live on metal-rich soils, and in the Philippines we have a lot of those. So I just want to put things into context. So what are metallophytes? So plants that, as I said, live in metal-rich soils, and they have adapted and are tolerant to elevated levels of heavy metals in soil. Oops, excuse me. Well, just to put also to an understanding, metals are important in plant nutrients if they are present in low concentrations. A lot of biochemical processes in a plant are co-mediated by copper, nickel, and zinc and iron, for example. And they're usually in very trace concentrations of usually 1 ppm, 1 microgram per gram. So if you like, that is 1 atom in a glass of water. But in high concentrations, metals are very toxic. So I just want to show you some of the key processes that are involved in a way in which metals can be taken up by a plant. So if you look at the bottom of that diagram, metal is acquired by a plant through its roots because the plant roots secrete organic acids, for example, which essentially dissolve the metals, and so the roots of the plants are able to absorb it and take it up. And then if you look at the left-hand side where I have chelation, what happens is these organic molecules that are present in the plant essentially wrap around the metal, and then they transport it up to the leaf tissues. So the benefit of this is it minimizes the toxic effect of an incoming metal, because metals, ionic metals, produce oxidative stress. So what happens once the metals are transferred by translocation, if we go to number 3, it is accumulated in the plant's tissue and in what are called cellular vacuoles. So if you like, they're little balloons inside plant tissues where the metal in liquid form is locked up and essentially with all these organic ligands. However, this is only in a very few species, because if you look at number 4, we have what is called phytostabilization, whereby most of the metals that are present in the soil are just stuck to the surface of the roots. So they're stabilized, and most plants that, if you like, live in heavy metal concentrations that are toxic do not take it up. So in one case with some of these plant species, they are very rare. Some species are what we call hyperaccumulators. These are what we can also colloquially call metal-eating species. And these are extremely rare. They only, to date, we have only discovered 550 plant species of flowering plants and a handful of non-flowering plants, which can accumulate metals, or hyperaccumulate in this case. And so these species are known to hyperaccumulate metals such as nickel, which is the most common one, copper, cadmium, zinc, manganese, lead, and thallium, and the metalloids such as arsenic, selenium, and antimony. And it is very rare that a plant will accumulate more than one metal or a semi-metal. In the ferns, you can actually hyperaccumulate arsenic and antimony, for example. And it is very curious that most species that can hyperaccumulate nickel, which is the most common metal that can be hyperaccumulated, belong to the cabbage family, the Brassicaceae. But don't worry, because in the Philippines, for example, there are very few of these species. Most of these Brassicaceae are actually herbaceous or soft tissued plants that live in the northern hemisphere. So, something happened. Excuse me. So what has this all got to do with the Philippines? I just want to take you on a little bit of a historical trip. So in the early 70s, a group of scientists from New Zealand, from the Massey University, led by Professor Robert Brooks, decided to do an important geobotanical survey of plants from the Southeast Asian region. Well, Robert Brooks and his colleagues had the hypothesis saying that, well, if plants grow on metal-rich soils or nickel-rich soils, which are present in the Philippines, so as you can see that diagram, the Philippines, Indonesia, Sabah, and New Guinea have very high concentrations of nickel-rich soils, or the ultramafics. Ultramafic means these are soils which have very high iron and magnesium, very low in calcium. And so this is a challenge in terms of plants growing on relatively toxic conditions. So Robert Brooks requested pieces of plant samples from herbarium from all around the world, mainly from Holland and the United States, which had collections from these places, and they analyzed them for the presence of metals. And so this is what happened. So the first report of Filipino plants showing heavy metal accumulation was demonstrated by Robert Brooks, and they showed two species of Flacourtiaceae, Homalium bittingensis and Homalium panayanum, which had elevated concentrations of nickel. These are not hyperaccumulators, but they're higher than usual, because anything above 100 ppm of nickel usually in a plant tissue signifies that the plant is being poisoned. However, what was exciting was the discovery of a tree species called Rhinerea bengalensis. This is a tree violet, which is quite well distributed along Southeast Asia and Australia, and also in the Andaman Islands just off India. And they discovered that this species of tree violet actually had concentrations of nickel which are really off the planet. There were more than 1%. And if you see there in the Philippines, Robert Brooks was really amazed that the Filipino species had more than 10,000 ppm, 1% nickel. And this is really quite remarkable. So what happened then after these late 70s, there were efforts to do some field studies in the Philippines. So in 1986, an English expedition studied the vegetation of the ultramafics of Mount Bloomfield in Palawan. So Mount Bloomfield is the mountain that you see when you go to the Puerto Princesa underground river. So from Sabang, the mountain on the background is Mount Bloomfield. So it was led by the ecologists John Proctor and Alan Baker. So Alan Baker was my PhD supervisor when I undertook my PhD studies 20 years ago. And so this is where I was introduced to the amazing world of Filipino hyperaccumulating species. So that's the view from up the top of Mount Bloomfield. I had the opportunity to go there in January 2019, retrace Alan Baker's footsteps and see for myself what it actually looked like and where some of these ultramafic species of hyperaccumulators are. So Baker and Proctor were able to discover and study the ecosystems, and they were able to identify 4 new hyperaccumulators. This was the first discovery in the Pacific, really. And so they discovered Euphorbiaceae, Meliaceae, and Ocnaceae, and Dicapetaliaceae. And if you look at those pictures there, if you go there now and retrace the footsteps, you will still see the remnants of the mining camp. People were mining for chromite and nickel, and you can see that huge scar on the side of Mount Bloomfield. But what was so exciting about that study was one particular species. So they discovered what they called Phyllanthus palawanensis, but then they asked a botanist to, to describe it. So the formal description, if you see, was produced 20 years later. But it was so, Baker and Peter Hoffman and Dr. Domingo Madulid, formerly of the Herbarium, National Herbarium, did a formal description of it. And it was dedicated to that man on the green shirt, Max van Balgoy, who was from Sabah. And he is a remarkable botanist. He has an encyclopedic knowledge of most of the flora of Southeast Asia. But so they named that species Phyllanthus for Max, so Phyllanthus balguia. What was so remarkable about it, its leaves contain 1.6% nickel, and the stems would also range from 0.5% to about 2 to 3%. And this was easily shown by a chemical indicator called dimethylglyoxime, which if you squeeze, crush some of the plant tissue, it will exude nickel, ionic nickel onto the leaves, and it will produce that quite dark red reaction. So that indicates that it is possibly a nickel hyperaccumulator. So if you cut the stem of that, what really amazed us, what you could literally have 20% nickel in the phloem sap. So it really asks the question, what are the metabolic mechanisms that allow it to tolerate so much nickel? I mean, to put things into context, our cutlery or our watch bands, for example, are usually about 8 to 15% nickel. This plant has liquefied nickel and tolerates putting it in itself. So this is where I come in, because in 2009, I had the opportunity to do a Balik Scientist under the invitation of the environmental scientists from Ateneo de Manila. So we went to one of these ultramafic regions in Zambales, and because I said, well, let's go there and have a look, see if we can find some nickel hyperaccumulating species. So this is some images of what it looks like. A typical ultramafic stunted vegetation with a lot of grasslands. And incidentally, that grass that you see there is quite endemic in the Southern Hemisphere. In Australia, we call that kangaroo grass. Dantonia, sorry, not Dantonia, it slips me at the moment, but I will tell you later. It escapes the name of what kangaroo grass. I think Demi will be able to help me jog my memory. But so what happened is we collected several species, which I had a hunch could be possibly hyperaccumulators. So of those 10, one was expressed in the dimethylglyoxime test, as you see there. I took some samples back to Melbourne, and we did the analytical work, and we demonstrated that it would go over the 1,000 parts per million level of nickel in the dry leaf tissue. So we consider 1,000 ppm as a good operative definition for metal hyperaccumulation. So I told the team in Ateneo, go back there after the end of the rainy season and test these plants again to see whether nickel uptake is much more elevated. So we demonstrated there is much more nickel once the plant is if you like, in more less dry conditions, if you like. So this was quite fascinating, but it begs the question, and this is a good study that has to be done in looking at how water availability regulates the accumulation of metals. And this is a project waiting to be done. So I just want to point you out to the recent work on localization of nickel by this Brainius Sonua, and this has been completed by, once again, the team in Ateneo de Manila. This requires a lot of intricate sectioning, and so this was done with the aid of light microscopy and electron microscopy. So if you look at those light images, you can see how the distribution of nickel in the cross-section of a leaf tissue. And so we mainly look at the epidermal tissue, which are the ones close to the surface of the leaf. And so you can see those red spots. So the distribution of the red spots through light microscopy indicate that most of the nickel is in the epidermal and also the tissue that just below the epidermis, and there is very little of it in the vascular bundle. So this indicates that it possibly transports the nickel, as I showed in the model earlier on in the schematic diagram. And this was verified with the use of electron microscopy, where the ratio of nickel in the epidermal tissue is nearly 4 times more than the vascular bundle, because it's just transported and then allocated in the epidermis and some of the mesophyll tissue that just under the epidermis. So this is a very good study, which is the very first ultrastructure study of these nickel hyperaccumulators. So now I take you to my meeting with the UP Los Baños scientists. As you can see there in the background, there is the back of Dr. Asuncion Raimundo. She actually rang me up just sort of a few days before I was set to go back home to the Philippines, to Australia. And she said, why don't you come up to UP Los Baños? I want you to talk to us about hyperaccumulators, because there is one of the teams in Los Baños that has just received grant from the DOST to do some work on these metallophytes, but we need a bit more of a background. So I gave a talk there, and I was so pleased with the reception there. What wonderful questions and answers. And so, as you can see, the Edwina was very articulate and very passionate about his questions. So he and Dr. Kimado said, we would like you to work with us and learn about your experience. But they said the only catch was that they had no funding to support my involvement. I said, well, to me, this was an offer too good to refuse. So we wrote up another Balik scientist application, and I was successful to get that. And so that year I was hosted by Dr. Macandog in the Institute of Biological Sciences, and then we also worked together with the College of Forestry and Natural Resources. So essentially with the UP Los Baños team, with the forestry team, they had this project to look at the ultramafic areas of the Philippines, look at the ecology of these areas. So they had 4 study sites in Zambales, near where I went to with the Ateneo team, Samar, Palawan, and Surigao del Norte. And this is very important because in these unique rocky outcrops, these are 5% of the land surface of the Philippines. And so this is also very important in trying to understand the ecosystems before they essentially disappear. Because mining nickel mining is done in most of these areas. So very soon after we did that work, we published, and we were able to identify 24 new hyperaccumulated species. So that's the paper there. And I just like to show you some photos of some of the species. Essentially, we come across a nickel accumulating forest. So that is one of the trees there with 1000 ppm. But we also had a very small herbaceous species of Phyllanthus, just like what Baker and Proctor discovered. But this is a very small one. But it really had these remarkable levels of 1.7%. However, we were excited when Edwino was able to find a new species of tree violet. It's a smaller, it's a tall shrub, small tree, and it is new to science. It was called Rhinerea nicolifera. And it was also one of these phenomenal hyperaccumulate, which we call hypernickelophores. So that's just the nickel glyoxine reaction. And so the work that Edwino has done is very important because it helps us understand how to restore large-scale open-cut nickel mines, which are distributed all around the Philippines. So the understanding we have done is vital for this, because in many ways it is not difficult to revegetate these areas, because these hyperaccumulators are also very tough species, but all the other species are also very important. So one of the spin-offs from this kind of work is trying to understand the metabolomics, or the metabolic pathways that are involved in nickel hyperaccumulation. So I just want to quickly show you some of the work in progress. So we have started trying to characterize that through chromatographic methods. So if you look there, we've started comparing 4 of the species and look at the different distribution and the amount of the different organic acids, possibly enzymes that can be extracted from these plant tissues. And so we're trying to understand what is important in nickel uptake. And perhaps some of these have some very important potential for pharmaceutical purposes, and also to understand, well, how does a plant tolerate such large issues of oxidative stress, and perhaps we can understand that in looking at some of the other types of degenerative diseases that we are faced as humans. So now I want to go to the work done on arsenic. So this is actually arsenic and copper. So with the Ateneo team, they said, you know, wherever we go, we notice that there are some fern species that are growing in disused and sort of in the mine waste from gold and copper. So up in the mountain province and also south in Mindanao and Surigao. So they found that there was one particular species of fern that was growing very successfully in these places. So what happened was this fern, which is called Pteris melanocaulon, was growing in the presence of 2 other known arsenic hyperaccumulators, Pteris vittata, which was discovered by our Chinese colleagues in 1998 by Dr. Chaoyang Wei, who worked with me in Melbourne University for 6 months, and he introduced me to these incredible plants. And then also of Pterogramma calomelalos, which was discovered as an arsenic hyperaccumulator by the people from Thailand, from Mahidol University, approximately 18 years ago. So we also had a very active collaboration with these people. But what was interesting is this Pteris melanocaulon that the Ateneo team had discovered was prolific, but when we initially tested it, there was very little arsenic present in its tissue. But I said, look, it doesn't mean that it might not be an arsenic hyperaccumulator. We have to learn how to propagate it. And I want to draw your attention to the technical assistant, Mary Jane Apuan, who was an excellent technician, because she was instrumental in making sure that we could effectively propagate, mass propagate the Pteris species and actually do the dosing test to see whether it will accumulate arsenic. So if you look at the table above, when you increase the dosing of arsenic, in this case we use sodium arsenate, what happened was the accumulation of arsenic really increased. And so once we get over the threshold of 1,000 ppm of arsenic, then we can demonstrate that this species is an arsenic hyperaccumulator. So very good controlled work. And so we are happy to say that, well, we finally cracked it in the Philippines. We can do some mass production of these arsenic hyperaccumulators. And I think Dr. Claveria will talk about it later on and how we actually do it. So one of the spin-offs is that we can actually think beyond the box, because arsenic in plant tissue could, you know, the Chinese have started doing research where they're looking at the possibility of using this arsenic as a form of treatment for leukemia, for example. So they're doing some very interesting work there. So lastly, I want to go to the work that we have been doing in Melbourne University. When I was in the Philippines, I asked around and said, well, if there are any young faculty or students doing their masters who would be interested in doing a PhD with us in Melbourne University, please talk to me, and we'll see if we can take it forward. So I was happy to say that we had an excellent candidate. So in that picture there, that's a picture of Hannah at her graduation a couple of years ago. And not only did she complete a wonderful doctorate, she was able to meet her husband. And so a wonderful thing is Hannah and Fernando, who comes from Ecuador, Fernando is also an excellent young scientist, have now been gifted with their first child, Rafaela, there. So it was such a joyful opportunity. But now going back to what Hannah actually did. So I asked Hannah, well, are you ready to do something which is very challenging, which is to look at the mechanisms of uptake of mercury and possibly gold in plant species, because there could be some very interesting studies involved in that. Hannah had done her master's looking at the phytotoxicity of mercury in some herbaceous species, so she was well equipped already. So Hannah did her research work on looking at different candidate species, but we were interested particularly in some species which had the potential to accumulate gold. And so we used cassava as our model plant. Well, you might ask, why is cassava very useful? Firstly, cassava is nasty because we know that cassava produces cyanide, and it is through the cyanogenic sugar called linemarine. And so cyanide is released when a plant is, well, leaf tissue is crushed, or if you break the roots of the cassava, and the cyanide obviously is produced in high concentrations and be quite toxic. So we thought beyond the box. If it can produce that much, I'm just going to take you to this. Because, well, cyanide is used for extracting gold from low-grade ore in the mining industry by converting gold to water-soluble coordination complex. So what we wanted to show, well, if it produces so much cyanide, does it actually function if we expose it to toxic levels of gold and mercury? So one experiment that Hannah did, where she was looking at identification of the linemarine through cross sections of root tissue. And if you can see there, the red coloring there indicates that the linemarine mechanism is functioning. So in other words, it is predisposed to be able to dissolve gold. So this is very important, and what she demonstrated, I mean, this is a very complex technical method, but she was able to show that the mechanisms are there to extract gold. So she then used an X-ray system called PIXI, proton-induced X-ray emission, to essentially map the distribution of the metals and all other elements that are necessary for functioning of plants. So if you look there, the top G, H, and I, you will notice that when plants actually accumulate, or any tissue that has nanoparticles of gold will turn violet, because nanoparticle gold is not actually gold colored, it's pink or violet. And so in her experiments, already by doing light microscopy, you could already see that there is gold there. And so with the PIXI method, you could see that those yellow spots that are around the epidermis and in the cortex are distributions of gold. So we had to ascertain whether this was real gold. So with X-ray diffraction through our colleagues in Bulgaria, we were able to demonstrate that if you look at those particles there, the more gold that is in solution, in the hydroponic solution, the more gold forms those nanoparticles. So there is an interesting mechanism in the dynamics of formation of these nanoparticles that we have to understand much more. But what was important is we were able to identify that these gold particles are pure gold nanoparticles. They are not amalgam of gold and mercury, which really was quite a remarkable thing. So in other words, in order to accumulate gold, you don't really need to actually spray cyanide into the soil. A plant which is cyanogenic, just like cassava, will do it for you. So this really creates a window of opportunity for some very high cutting edge research, and I think Hannah has pioneered this, and you know, it starts an opportunity for us to take it one step further. So I just want to finish with that, and by summarizing the work that we've been done over the last 10 years. So the Philippines and Brazil are jointly the third major biodiversity hotspots for nickel hyperaccumulators. As of yesterday, I was speaking to Dr. Quemado and Dr. Fernando, and they said that they have identified a new one. So this brings us to a total of 37. Saba, which has been worked on quite a lot over the last 10 years, has come up with 24 species. So in this part of Southeast Asia, we're not very far off in really going to the numbers which have been discovered in Cuba and New Caledonia, where most of the tropical nickel hyperaccumulator species have been discovered. So we have started work on the phytochemistry. The first phytochemistry study on hyperaccumulators has been done in New Zealand from powdered leaf tissue, which was sent there. But now I think we have the capacity to do it in the Philippines. So the first publication of nickel localization in a native hyperaccumulator has been demonstrated. So to now we have 7 peer-reviewed papers on nickel. And then with the arsenic work, we have 6 peer-reviewed papers on arsenic and copper. And then on the work on gold and mercury, we have 4 peer-reviewed papers. So we have a combined output of 17 papers in 7 years. And Dr. Claveria has just notified me that there are 2 more that have just been accepted for publication. So I'm very pleased to say that it's been a wonderful output. This collaboration has been quite remarkable. So I just want to end with that. Thank you very much.
Thank you so much, Dr. Doronilla, for that very wonderful and informative talk. I think it's really important for us to have a lot of collaborations with other scientists as well, because that increases our opportunities to, move on and get more funding and be able to work on more things, diba? Ika nga nila, bayanihan really works well. Okay.
Yes, I suggest that, well, thank you very much for your patience. We've certainly been listening for an hour and a half. So I suggest that we have a 5-minute breather, and then we'll get back to the second half of our session.
Mm-hmm. Are we going to do the Q&A session before,
oh, yes, please.
When we come back? Or you want to do it now?
Well, we might as well do we can give 5 minutes to Q&A, yes. Thank you, Jen. So you can type your questions if you like.
Mm-hmm.